Hi, everybody. Welcome to the Chapter 10 review. This is going to be going over microbial metabolism and how cells basically make their energy. Remember that metabolism is referring to the total sum of all of the reactions, chemical reactions that would be going on in a cell, whether we're building or breaking down, right? So if we're breaking down, we call that catabolism. Remember, I refer to it as I remember as cats are destructive. And so my cats would be breaking down our molecules, and that would be releasing energy that we can use to drive anabolism, which would be building new things. Now, if you're building a building, do you need energy to build the building? Just literally, do you? Yes, of course you do. You can't just not have some sort of energy, and the bigger your building, the more energy you're going to need probably. So that's the idea. You need energy to drive building new things. That's anabolism. You got to have energy for it. We get that energy typically through catabolism, breaking things down, and as we break it down, using the energy that is left for our benefit. And the way that those two work together, we call and rely on one another and create intermediates that allow us to make new molecules and new steps from all these branching pathways that we have going on. That is called amphibolism. It is literally amphi, and then like catabolism or bolism. Okay, so let me write it. I guess we can write it out for you because you like words to be spelled probably. Otherwise, it's just a sound. Okay. Okay, so that is basically working together, the catabolism and the anabolism, and then relying on one another. So how is all of this driven? A lot of this is driven via enzymes, and enzymes are catalysts that lower the activation energy of reactions without being used up as a reactant itself. What does that mean? It means there's no permanent change made to an enzyme. So it's usually a protein. There's no permanent change made to that enzyme when it is acting on its substrate, which is the thing that it is going to convert into the product at the active site, right? So the shape of that enzyme is very, very important in how it interacts with its substrate. It's very specific as well. And that happens again at the active site of the enzyme. That helps speed up reactions and helps overcome the amount of energy that would necessarily be needed to be added in for the reaction to occur by itself. Essentially, enzymes are going to help reactions happen at what I like was cutely called the speed of life. I like that term. So that's what enzymes are going to allow for. So enzymes, we know they act on a substrate at the active site. That's where they're going to interact with it. They often can require a cofactor. And that is something that it has to interact with in order to function. That can be a metallic ion that's associated with it, like iron or zinc or copper or something. Or it can be an organic molecule. So typically that would be something like so it's organic, non-protein. That would be something like a vitamin, like vitamin B12 or something. Those can act as coenzymes when they are organic molecules. Those are important, and the availability of those can help regulate the ability of the enzyme to work. You can also regulate the ability of the enzyme to work at the genetic level. So if there is any need to make a protein versus always producing a protein, controlling that would be constitutive versus a controlled sort of enzyme. Then yeah, you can do that at the genetic level. You can also do that at the enzyme level. So what we have at the enzyme level, we have competitive inhibition versus non-competitive inhibition. Competitive inhibition is we're referring to the inhibitor, whatever's inhibiting the function of your enzyme. It binds at your active site where your substrate would have been acted on, except the enzyme can't act on the inhibitor. And so it'll keep trying, but because it's not being converted into I helped you now mode, essentially changing its shape or something, it's going to keep trying. And so it doesn't, it can get stuck that way, and that makes it unavailable for the substrate, and the substrate therefore cannot be acted upon. And that helps, prevent that from happening if you need to prevent it. So that's competitive inhibition. Non-competitive inhibition is whenever you have usually the product of whatever's happening. So if you are enzyme A and your job is to convert A into B, you're, as an enzyme, going to bind with A substrate at your active site. And instead of, so you're going to convert A into B and keep going like that. And you have enough B now made up, just a whole bunch of B hanging around. And B can interact with another portion of that protein called the regulatory site or the allosteric site. It's not the active site. Okay, completely different site, different part of that protein. But whenever that product, when B interacts with enzyme A, it's going to cause a change in the shape of enzyme A, so that now the substrate of A, which would get turned into B, cannot bind in the active site because we changed the shape. No longer can A fit into that active site spot. So that's non-competitive because we didn't compete for the active site. So definitely know the difference between those two. Stepping a little bit back on the slide, we talked about the different kinds of enzymes. We have the enzyme classes we have 6 of them: oxidoreductases, transferases, hydrolases, liases, isomerases, and ligases. I would be aware of what each one does, but oxidoreductase is going to deal with transfer of electrons from one molecule to another. Definitely know what it is to be oxidized. If you lose electrons, you have been oxidized. If you gain electrons, you have been reduced. Definitely understand that whole system and how that works. It's a very important concept for this chapter. The different enzyme classes, oxidoreductases, those are the ones that are going to be involved in electron transfer. We had talked about how oxidation is loss of electron, and reduction is gain of electron. So you can talk about molecules as being reduced or oxidized based on whether they gained electrons or lost electrons respectively. So that's what oxidoreductases are going to be involved in. They're the enzymes involved in reactions that involve electron exchange. Next, we have the transferases that remove and transfer functional groups from one molecule to another. Then we have the hydrolases. This is going to be adding water in order to actually break bonds. So that would be like hydrolysis, which would be breaking down of molecules by the addition of water. Then we have liases that involve double bond formation. Then we have the isomerases that have to deal with the structural alteration of a molecule while still maintaining its overall molecular makeup. And then we have ligases, and this is going to be like DNA ligase, and this is involving creating bonds in molecules. Like we were creating the phosphodiester bonds that were to be found in DNA when we needed to join together, like our Okazaki fragments or something like that, little fragments of DNA. We have to ligate them back together using DNA ligase. So those were our 6 types of enzymes. Then we talked a little bit about denaturation of proteins and importance of the shape of a protein. Any sort of effect on the protein that leads to the change in the protein's shape that causes that protein to no longer be able to function because of that, we would call that as denaturation. That can be caused by heat. That can cause any hydrogen bonds and associations between hydrogens and charged molecules to be dissociated, as well as pH. If we have a large change in pH from what that protein normally functions at, that can cause a change in the hydrogen bond interactions with polar molecules across the way. So Anyways, that would cause detrimental changes to the shape of the protein and therefore the function of the protein. So that's how denaturation operates. So that's that for that portion. The next portion of the chapter had mostly to do with actual energy production. And in the cell, our main energy molecule is going to be ATP, adenosine triphosphate. It's made up of a nitrogenous base adenine, 5-carbon sugar ribose, and 3 phosphate groups that are highly negatively charged and are going to be pushing apart from one another and causing tension. And all of that is creating energy that just wants to be released by removing those phosphate groups. So that's how the energy works. The number one way for cells to make energy would be through aerobic respiration. Cells can also undergo anaerobic respiration. And if they're anaerobic respiration, they're using anything except for oxygen as their final electron acceptor. They can also undergo fermentation. This is mostly for facultative anaerobes as well as aerotolerant anaerobes. Fermentation is not going to create anywhere near as much ATP. They're going to be using the organic molecule intermediates as a means to be a final electron acceptor, so we can keep going back through glycolysis. And that creates the formation of alcohol and or acids as byproducts of those. So it just depends. So that's fermentation. And we now should probably go ahead and talk about aerobic respiration. So aerobic respiration has 3 main steps: glycolysis, Krebs cycle, and the electron transport chain. In glycolysis, we are breaking apart glucose, which is a 6-carbon sugar, into 2 3-carbon pyruvic acids. So we make 2 3-carbon pyruvic acids. Each one can be converted into acetyl-CoA, and each acetyl-CoA can enter into the Krebs cycle. So we have 2 runs through the Krebs cycle for each glucose molecule. Basically, when we enter into the Krebs cycle, we are going to start removing carbons and harnessing the electrons that are going to be involved in that removal, so released in that removal, and using those electrons to make our electron carriers carry electrons. So that's NAD and FAD, and those guys are going to become NADH whenever they're reduced, or FADH2 when it's reduced, respectively. So that's what's going to be going on with those electrons. Then we make carbon dioxide as a byproduct. So we are going to be releasing that, exhaling that as a waste product. And that's where the carbons are going to go from the glucose molecule. We're just wanting the electrons, really. That's because we want to put the electrons down the electron transport chain, which is the 3rd step. So those electrons on the electron carriers, NADH and FADH2, are going to be shuttled through the electron transport system. There are a series of proteins called cytochromes, and they're essentially going to shuttle the electrons and be excited by the electrons as they're being shuttled through each one. And as they're moving the electrons through, and then you have that negative charge of that electron moving through each of these cytochromes, we will pump hydrogen ions, positive charge hydrogen ions, across the membrane to create the proton motive force. And this whole thing where we have the movement of the electrons across, driving the pumping of the hydrogen ions out on the other side of the membrane, this is called chemiosmosis. That's the term for it. Anyways, we made our proton motive force. It's got a nice positive charge outside of the membrane. And now what we're going to do is allow them to come back into the cell, because now they want to come back in. We've got all these hydrogens outside of the cell positively charged. They want back in to equal everything out on both sides of the membrane. So they come in through ATP synthase, and when they do that, it essentially drives the phosphorylation of the ADP into ATP. This is creating ATP via oxidative phosphorylation, because it's driven by that whole electron transfer spiel. Yes, so as a result of all of this happening, we now have electrons at the end of the chain that need to be gotten rid of, and we have protons that got pumped into the cell that need to get gotten rid of. So we take those H+, and give each of them electrons, and there's H and H, and then pop those onto an O from an O2 that we breathed in, and that creates water as a byproduct. So we've taken care of those electrons and protons. So that is how that's taken care of and how we end the production of energy. And of course, this is constantly going on. It doesn't just happen like once and then that's it. Anyways, for bacteria and archaea, this whole aerobic respiration process is going to occur along the inner membrane or the cytoplasmic membrane, essentially, of the cell envelope, whatever, if you're gram-positive or negative, whatever you are. And then in eukaryotes like you and I, this is going to be happening on the folds of the cristae of the mitochondria in our cells. Then we already talked about amphibolism and how that's catabolism and anabolism working together. We talk about that. And we did talk about fermentation, and that's only going to happen whenever oxygen is not available. We have facultative anaerobes that whenever oxygen is available, they're going to undergo aerobic respiration because it creates more energy. So of course they're going to do that. However, if there is no oxygen, they have fermentation available as a means to create energy for their cells. Then we talked about photosynthesis a bit. This is referring to the light cycle and the dark cycle. The light cycle literally involving light exciting, pigmented molecules, essentially allowing us to create the pumping of the proton across the membranes of the chloroplast or the cell, if you have bacteria. And when that happens, again, proton motive force can allow for ATP synthase to be activated, and we can make a TP from a DP due to that. So that's driven by the light. So when we have that ATP built up, we can now use that to essentially take the carbons from carbon dioxide from the atmosphere and create organic compounds like glucose in the Calvin cycle, or the dark reactions of the plant cell or the bacterial cell. And they would need to do that to create, of course, molecules that they need for building new cells, but they also need to do that to create their own energy, because they also use mitochondria and aerobic respiration to drive their reactions in their cells. So yeah, so that's basically how that will work. So that is chapter 10 in a nutshell, and I will see you guys for the next one.